In 2020, an AI system solved a problem that had defeated the world's best scientists for 50 consecutive years. And it did it in a matter of days. The score it achieved, 92.4 out of 100, was so far beyond what anyone expected that the scientists judging the competition thought their scoring system had broken. But here's the question that nobody has fully answered. How did a team of engineers with no biology degrees crack something that Nobel Prize-winning biochemists had spent entire careers failing to crack? And what does it mean that a machine got there first? That answer will change how you think about medicine, about AI, and about the future of human life itself. To understand why 2020 was so extraordinary, you need to go back to 1972. That year, a biochemist named Christian Anfinsen stood in Stockholm and accepted the Nobel Prize in Chemistry. His discovery was deceptively simple. The shape of a protein is determined entirely by its sequence of amino acids. The sequence is the code. The shape is the result. Simple in theory, catastrophic in practice. Because figuring out that shape, actually predicting the precise three-dimensional structure a protein would fold into, turned out to be one of the hardest computational problems in the history of science. Here's why it matters. Proteins are the machinery of life. Every cell in your body runs on them. Your immune system, your metabolism, the way your neurons fire, all of it depends on proteins doing their jobs. And proteins only do their jobs because of their shape. A misfolded protein doesn't just stop working. It can cause Parkinson's disease, Alzheimer's, cystic fibrosis. The shape is everything. Scientists knew the sequence. They couldn't get the shape. And the gap between those two things, that gap, became known as the protein folding problem. For 5 decades, the best minds in biochemistry attacked it. They used X-ray crystallography, nuclear magnetic resonance imaging, cryo-electron microscopy. Each method took months, sometimes years, and cost hundreds of thousands of dollars per protein. By 2020, after 50 years of effort, scientists had mapped the structures of roughly 170,000 proteins. There are an estimated 200 million proteins in nature. The math on that is brutal. Enter Demis Hassabis. He was 23 years old when he finished his undergraduate degree in computer science at Cambridge. By the time he was 26, he had a PhD in cognitive neuroscience. He wasn't a biologist, he wasn't a chemist. He was a chess prodigy turned neuroscientist turned game designer who genuinely believed, that artificial intelligence could solve problems that human intelligence couldn't. In 2010, he co-founded DeepMind Technologies in London, alongside Shane Legg and Mustafa Suleyman. The pitch was audacious. Build AI that thinks like a scientist, not AI that plays chess. AI that does science. For 4 years, they ran on ambition and limited funding. Then in January 2014, Google wrote a check for $600 million. $600 million for a company with no products, no revenue, and a mission that most of the tech industry considered borderline delusional. But Hassabis had a plan, and within 4 years the world would get its first hint of what that plan looked like. In 2018, DeepMind entered a competition called CASP, the Critical Assessment of Protein Structure Prediction. Scientists had been running CASP every 2 years since 1994 as a benchmark, a way of measuring how close the field was getting to solving the folding problem. For 24 years, progress had been slow, incremental, grinding. AlphaFold, the first version, won the 2018 CASP competition by a margin that stunned the field. But the 2018 result was just a warning shot. Because what Hassabis and his team were building in secret, what they were preparing to unleash at the next competition, was something the scientific community was completely unprepared for. Remember the question from the opening. How did AlphaFold crack something experts thought would take another 50 years? We're getting there. But first, you need to understand what made the second version so different. After the 2018 win, Hasabi's brought in a researcher named John Jumper. Jumper had a PhD in theoretical chemistry from the University of Chicago, and a background that sat precisely at the intersection of physics, chemistry, and machine learning. He wasn't just a programmer. He understood the biology at a molecular level. Jumper looked at the first AlphaFold and saw its ceiling. The original system treated protein folding as a pattern-matching problem. Feed it sequences, find patterns, predict structure. It worked better than anything before it. But it had limits. What Jumper built for AlphaFold 2 was fundamentally different. He designed a neural network architecture called the evoformer, a system that didn't just look at a single protein sequence in isolation. It analyzed how that sequence had evolved across millions of years of biological history. It looked at thousands of related proteins across hundreds of species and asked, where did the mutations happen and where didn't they? Because when two positions in a protein sequence always mutate together across evolutionary history, that's a signal. It means those two positions are physically close to each other in the folded structure. They co-evolved because they have to touch. The evoformer turned 3.5 billion years of evolution into a dataset. Stop. Before we go further, you need to understand something. This wasn't just a better algorithm. This was a completely different way of thinking about the problem. Every other approach in 50 years of research had tried to simulate the physics of folding, to model the forces and energies that pull a protein into shape. AlphaFold 2 didn't simulate physics. It read the evolutionary record and let billions of years of natural selection tell it the answer. The team worked through 2019 and into 2020. The CASP P14 competition, the 2020 edition, was held virtually in November because of COVID-19. The timing was almost poetic. The world was in the grip of a pandemic caused by a misfolded spike protein on a coronavirus. And here was a machine that could predict protein structures with atomic-level accuracy. When the results came in, the scientific community went silent. AlphaFold 2 scored 92.4 on the GDT scale, a metric where 100 represents a perfect match to the experimentally determined structure. The previous record, set by AlphaFold in 2018, was around 60. The average for all other competitors in 2020 was roughly 40 to 50, 92.4. For most proteins, the predictions were accurate to within the width of a single atom. John Molt, one of the founders of CASP and a structural biologist who had spent decades watching the competition, said publicly that this was a solution to the protein folding problem. Not progress, not a step forward, a solution. But the score wasn't the most important part of what happened next. The most important part was a decision that Hasabi's and DeepMind didn't have to make, and that almost nobody expected them to make. At this point in the story, DeepMind was sitting on something worth billions of dollars. Pharmaceutical companies spend an average of $2.6 billion and 12 years to bring a single drug to market. A significant chunk of that cost sits in the early research phase, figuring out the structure of target proteins. AlphaFold 2 could potentially collapse that phase from years to days. The business play was obvious. License the technology, charge pharmaceutical companies for access, build a proprietary database, turn the 50-year scientific breakthrough into a $10 billion revenue stream. That is not what happened. In July 2021, DeepMind released the AlphaFold protein structure database, completely free to every researcher on Earth. The initial release contained predicted structures for 350,000 proteins. including the entire human proteome, every protein the human body makes. Within a year, they expanded it to over 200 million protein structures, the entire database. Free. Open access. This is where the open loop from the beginning closes, because this is the part that explains why AlphaFold's impact went so far beyond what anyone projected. Within 12 months of the database launch, more than 500,000 researchers in 190 countries had accessed it. Scientists in Kenya used it to study malaria proteins. A lab in Brazil mapped dengue fever targets. Researchers at Oxford used it to accelerate work on antibiotic-resistant bacteria, a problem the WHO had declared a global health emergency. The $1 billion figure you hear cited as potential pharmaceutical savings? That's almost certainly an undercount. And it's already outdated. But here's what no one saw coming. The open release created a scientific arms race, but in the right direction. Because once every researcher on Earth had access to 200 million protein structures, the questions they could ask exploded in scope. Scientists weren't just using AlphaFold to find drug targets. They were using it to understand how viruses hijack cells, to design enzymes that eat plastic, to map the molecular basis of aging. In 2022, a team at the University of Washington used AlphaFold data to design entirely new proteins that don't exist in nature. Proteins engineered from scratch to perform specific functions. This was the next frontier. Not just reading the protein code, writing it. The conflict, when it came, wasn't a corporate battle or a regulatory crackdown. It was quieter and more fundamental than that. Some researchers pushed back on the open database itself, not because they opposed sharing, but because they worried about the accuracy of predictions for proteins in unusual environments, or proteins that interact with other proteins in complex ways. AlphaFold 2 predicts single protein structures with extraordinary accuracy. But biology is messier than that. Proteins don't work alone. They bind, they interact, they change shape depending on context. Hassabis acknowledged this directly. In interviews, he was careful to say AlphaFold was a tool, not an oracle. It solved one version of the problem. The deeper version, predicting how proteins behave in living systems, under stress, in disease, that problem remained open. And then, in October 2024, the Nobel Committee made a decision that settled the historical argument once and for all. John Jumper and Demis Hassabis were awarded the Nobel Prize in Chemistry, alongside David Baker of the University of Washington, who had done foundational work on protein design. The Nobel Committee cited AlphaFold explicitly as having solved a 50-year-old problem. It was the first time in Nobel history that a machine learning system was central to a chemistry prize. And it raised a question that the scientific community is still arguing about. If AI can solve problems at this level, which problems are we still treating as unsolvable? When the answer is already within reach, here's where we are today. The AlphaFold database now contains predicted structures for over 200 million proteins, the entire known protein universe, mapped in less than 3 years. What 50 years of experimental biology produced, 170,000 structures. What AlphaFold produced in 24 months, more than 1,000 times that. New drugs are moving through development pipelines that wouldn't exist without AlphaFold data. At least 3 major pharmaceutical companies have publicly credited AlphaFold with accelerating early-stage research by 18 to 24 months per project, at $2.6 billion per drug, and 12 years per development cycle. Shaving 2 years off the front end doesn't just save money, it saves lives. The ones that would have been lost while the drug was still being developed. The plastic-eating enzyme story is worth pausing on. Researchers used AlphaFold to understand the structure of a naturally occurring enzyme called PTase, which breaks down plastic. They use that structural data to engineer a supercharged version that works 20 times faster at higher temperatures. The implications for plastic pollution: 380 million metric tons produced every year, are still being calculated. DeepMind itself has moved on to the next problem. AlphaFold 3, released in 2024, predicts not just protein structures but the structures of DNA, RNA, and small molecules, and how they all interact with each other. The complexity of that problem makes AlphaFold 2 look like a warm-up exercise. Demis Hassabis, now Sir Demis Hassabis, after receiving a knighthood in 2024, has said publicly that he believes AI will compress the next 50 years of scientific progress into the next decade. That's not a marketing line. That's a man who watched an AI system do in 2 years what biochemistry couldn't do in 50, and is now asking what else has been waiting. The deeper legacy of AlphaFold isn't a database or a Nobel Prize or a pharmaceutical pipeline. It's a proof of concept. It demonstrated, with a 92.4 GDT score and 200 million protein structures and 500,000 researchers in 190 countries, that artificial intelligence can be a scientific instrument, not just a productivity tool. Every hard problem in science now has to be re-examined through that lens. Climate modeling, materials science, neuroscience. If AlphaFold could crack protein folding, what else have we been treating as permanently out of reach, when the real answer is that we just hadn't built the right tool yet? That question doesn't have a comfortable answer. It has a motivating one. The 50-year problem is solved. The next 50 years just got a lot shorter. If this story changed how you think about what's possible, subscribe. We put one of these out every week. And next week, there's a man who, for 11 years, ran the most powerful financial operation in history, completely outside the law, and the government had absolutely no idea he existed.